Hello everyone, welcome to the first CIBM Breakfast and Science of 2026. It's great to see you all here, almost a full house, and I think we have many people online as well. And it's a great pleasure to introduce our speaker of today, Thomas Bolton. I'm going to give a quick overview of Thomas, although I'm sure everybody knows him since many years. So Thomas did his master at EPFL in life science engineering, and afterwards he was intrigued by research, but he didn't yet know exactly in which direction. So he went first into animal research, and that wasn't really his thing. And then he moved to computational side of things, and that was really his thing. So he did a PhD in my lab, and afterwards he went on to explore the Lausanne, Geneva area, let's say the Connectomic. So he was first a postdoc after the PhD with Constantin Tuliesk at CHUV. And then he moved to the lab of Patrick Hagman, which you all know also at CHUV, and where he still has some affiliation today, right?
Not officially.
Not officially.
Yeah, but the work I'm presenting today was partly carried on there, yeah.
And after that, he moved to the lab of Ingrid Beek here at University of Geneva, Campus Biotech, shifting a bit gears from graph signal processing to neuromodulation and TMS, although I think today the talk will be fully covering GSP.
Yes, correct.
Not TMS. But that's maybe for next talk.
Who knows?
And I like to mention also that Thomas has been very successful. He has many influential publications, and he also got awards for his work. In particular, for his PhD thesis, he got the Swiss Society for Biomedical Engineering Research Award back in 2020. With no further ado, please, Thomas.
Thank you, Dimitri.
Very honored to have you here today. Thank you.
The honor is all mine. Thanks a lot. Hi, everyone. First, just to mention that all the success and the setting on computational neuroscience partly goes also to Dimitri for having been such a great mentor and supervisor. So it's a great pleasure to be here today and to tell you about how to improve graph signal processing tools for brain behavior analysis. As Dimitri mentioned, I now work at University of Geneva, but the work I'll present today was conducted during my position with Patrick Hagman, and then I continued carrying it on the side afterwards as well. So first question, of course, would be why do we have to conduct brain behavior analysis? What's the point? We all wish, I think, to be able to diagnose brain disorders early and efficiently in robust manner. And of course, we would also all wish to be able to treat individuals suffering from brain disorders, ideally in personalized manner using underlying biology. A good start for this can be to try and predict behavior from neuroimaging features, because actually it's increasingly acknowledged that psychiatric symptoms and concomitant changes in brain function manifest along a continuum. Patients suffering from such disorders may just express some facets of brain function more strongly than healthy controls. And so it makes sense to use continuous approaches like prediction approaches in this setting, rather than the more traditional group contrast approaches, for instance, comparing controls to a population of patients. So in other words, the goal is to exploit the fact that there is a lot of inter-individual variability in terms of brain structure and function. How do we do this? We want biomarkers to satisfy several important conditions, starting with stability and individuality. Ideally, we want to measure a quantity that will not change over the course of time in a given subject, and we want that this quantity be different from a subject to the other. At the same time, we also want to measure something relevant, where a difference in our biomarker will also relate to a difference in the behavior of interest. So far, functional connectivity, the statistical dependence between regional activity time courses, has been the most influential feature used in such settings, and it has been primarily considered in the prediction of cognitive abilities. Just a very brief reminder of what functional connectivity is. In a very simple example, you see two regional activity time courses in blue and in red. And you see that in this case, the two regions activate and they activate together pretty well. You can plot this same data as an activity by activity plot and observe a clear positive valued slope. In other words, there is a positive correlation between the time courses, that is a positive functional connectivity. You can compute this for all pairs of brain regions and obtain a representation as a matrix, a so-called functional connectome, where each element in this matrix denotes the dependence between a specific pair of brain regions. Functional connectivity is stable and individual. As an easy example, it's possible to fingerprint subjects using functional connectivity, which implies that functional connectivity remains reasonably stable and is also different across subjects. And there have been extensive studies in order to try and predict different facets of cognition using functional connectivity. For example, here, I just show you some results from Thomas Yeo's laboratory from a few years back, where functional connectivity, the red box plots across two different data sets, the Human Connectome Project data set on the left and the ABCD data set on the right, enabled very high satisfactory prediction with R values up to 0.4 or even 0.5. Now, the goal is to go beyond functional connectivity and perhaps leverage graph signal processing as a framework to hopefully do even better. So let me briefly introduce what graph signal processing is. Graph signal processing includes a graph, a signal, and some processing, as the name indicates. And the graph is typically inferred from diffusion magnetic resonance imaging data. The first step is to pick a brain atlas, that is a parcellation of the brain into regions of interest. There is variability at this stage. There can be different types of atlases based on structural or functional markers, for instance. There can be different scales. The atlases may contain 100 regions, 500 regions, or even more. And the content may differ as well. Some atlases only cover the cortex, others include the subcortex and the cerebellum, which are also very important in the brain. Then, from the diffusion MRI information, using the atlas, we can go from information regarding the diffusion of water molecules to the computation of tracts that link different regions of the brain. And subsequently, we can partition them to quantify structural connectivity between pairs of brain regions. That is, to quantify the extent of physical wiring between different regions. And typically, people work with a property of interest. It can be the density of fibers linking to regions. It can be the length of the fibers. It can be fractional anisotropy. There are many different choices. Once one has set on a property of interest, the next question is whether to generate a structural connectivity matrix, a representation of this physical wiring at the whole brain level, only once for the whole group as an average across the subjects, or once per subject to be able to account for possible differences and for individual attributes. And I mentioned graph at first, but now I've been speaking about structural connectivity. In a structural connectivity matrix, each entry actually is an edge in the graph that I'm referring to, and each node of the graph would be a brain region. Now, the signal is typically functional magnetic resonance imaging derived. As you probably all know, fMRI enables to non-invasively track brain function, albeit in indirect manner as we quantify metabolic coupling. It is a somewhat sluggish and delayed response compared to electrical activity per se, but it's still very interesting. It covers the whole brain, and it enables us to derive regional time courses of activity using the same atlas as used for the generation of our structural graph. So for each region at hand, we are going to track metabolic activity over the course of time. And we can view each moment in time as a signal, a multidimensional signal of size, a multidimensional signal of size R, R being the number of regions at hand. You see an example at the bottom left of the slide where I depict a graph with nodes and edges. That would be your brain with regions and physical wiring between them. And then an example signal on this graph where each of the nodes has a certain value associated to it. That would be activity at a certain moment in time, and there would be one signal per moment in time. Now, what about the processing? for the sake of simplicity, we will just consider an average structural graph for the whole subject population. And I'm going to denote the structural connectivity matrix as capital A. I'm going to introduce some mathematics here, but for the members of the audience who are not keen on, let's say, getting into details for that, they will always be in these yellow frames, and you can just discard them and focus on the conceptual parts instead. For the mathsy people, feel free to follow along, of course. So the goal is to convert our structural graph into connectome harmonics, a means to efficiently summarize brain structure, as I will further explain. The recipe to do this, in simple terms, is to first compute the diagonal degree matrix of the graph, that I term D, and then compute what we call a graph shift operator. The graph shift operator basically informs you about the topology of the graph, and typically we use a certain variant of the Laplacian, for example. Here, I consider the unnormalized Laplacian, which is defined as the degree matrix to which you subtract the adjacency matrix A. If you perform an eigen decomposition of this graph shift operator, you get a matrix U and a diagonal matrix lambda. And each column of this matrix U is one of the eigenvectors and is also actually a connectome harmonic. You can see an example of this at the bottom of the slide, where I showcase a few connectome harmonics, a few eigenvectors performing this approach on data from the Human Connectome project. You can see that the first harmonics show very smooth fluctuations of signal along the surface of the brain. The first one is actually almost a flat signal. You can see blue everywhere. The second one contrasts both hemispheres. And as you progress in the hierarchy, you have a contrast that goes in increasing fashion between topologically connected brain regions. In fact, the more you progress along this indexing, the higher the eigenvalue, and also the higher the frequency conveyed by the harmonic, which you can see intuitively. Did I see a question?
Yes. I was wondering, in the surface plot,
yeah,
it's represented exactly in color. Is the average edge strength for each node?
Okay, so the color here represents the signal on the graph, that is the brain. It looks like, I mean, the tool that I used for this was a BrainNet viewer, and it basically converts, if you want, a regional signal where you have a uniform value in a given region into sort of a voxel-wise representation. So the conversion is not perfect. This is why you also see some slightly lighter colors. But basically, if you want, it's sort of an extrapolation of the regional signal. So when you see a blue value, it means that at that location along the brain surface, the signal was, in this case, negative valued. When you see red, it means that the signal was positive valued. And so you can see that for the first harmonics, when you progress along the surface of the brain, which also means that you jump from a given node to strongly connected nodes to that one, you have a very smooth changes in value. You go from dark red to bright red, but very slowly. And you see that if you look at the bottom harmonics, it's the opposite. You can go locally, jump from a spot to a neighboring spot along the surface, and you have a very big signal change.
Sorry, maybe, but I still miss it. So you go from edge to node, the conversion from edge to node. I mean, the matrix is edges, right? But the surface is node.
So the structural connectivity matrix is, as the name indicates, a matrix, but then each of the eigenvectors is a vector. Basically. So if you want, in the equations, A and L are matrices. Capital U is a matrix, but each column of capital U is one of the eigenvectors or one of the connectome harmonics. And so there is a number of elements equal to the number of regions, hence it's a signal on the graph.
So it's a value of,
yeah. And then as you progress from top to bottom, you consider eigenvectors linked to increasingly higher eigenvalues. At the same time, these eigenvectors show increasing spectral frequencies. You see that the oscillations, the change in signal between connected nodes in the graph become larger and larger. Is it clearer now? Yeah? Okay, great. Any other questions at this point? No? All right, so let's keep going. Now, about this processing, I've explained how to obtain connectome harmonics. And I want to make a parallel with Fourier analysis to explain how these connectome harmonics can be used to represent a time point of whole brain regional activity. So in traditional Fourier analysis, we have a signal. Here it's denoted in red, f of t, typically a signal over time. And it's possible to express this signal as a weighted sum of basis functions, which are basically complex exponential functions. You can see this process in the example over here. Mathematically, it basically means that to get our signal, we sum a bunch of weighted oscillatory functions. That's what it means. We can think of a similar approach in terms of signals living on the graph. We have a signal x living on this graph, and this is basically a weighted sum of our connectome harmonics, these oscillatory patterns, each weighted with a certain weight. In the gray frame, you see this equation as a sum, and now at the bottom right of the graph, you see the same equation in matrix form. This is a very important equation in graph signal processing called the graph Fourier transform. It tells you that if you have a signal living on the graph, x of t, you can express it as weighted combination of connectome harmonics. Conversely, if you want to infer spectral weights for a signal x of t, you can project your original signal onto your basis of harmonics. This is the forward graph Fourier transform. And when it becomes interesting is that you can do more than that. What you can do then is only keep a fraction of these weights and discard the rest. If you do that, you do what we call filtering. You only retain a certain contribution of the original signal as a function of the underlying structure. For example, you may want to retain only the fraction of the signal that is in line with the underlying structure, that is associated with the lowest frequency connectome harmonics, or perhaps to do the opposite. Now, let me go through a few studies that have addressed brain behavior prediction in the case of cognitive abilities and try to compare the specific choices made in each of these studies. First, I want to mention the work by Medaglia and colleagues, where the authors considered a perceptual switching task. You have a schematic of how the task works at the bottom left of the slide. You can see that in each case of a stimulus, there is a global feature, for example, a triangle. But the triangle is made of smaller shapes that are put together. In this particular case, it would be very small squares. And the subject is asked to give either the global shape or the local shape, depending on the color of the stimulus. So in the top example, the color is always the same. So you will always, for example, give the global shape, circle, square, triangle. But in the bottom, the color changes. So you need to think every time, okay, do I need to give the global or the local shape? And you take more time to answer. And you can compute the difference in doing these two things, which is called the switch cost, and use this as a measure of cognitive abilities in this case. They considered 28 subjects, and as an atlas, a concatenation of different available parcellations for a total of a bit more than 100 regions. The modality that they considered was streamline density, and they worked with an average population-wise structural connectivity graph. The graph shift operator that they used was directly the connectivity matrix, which is a possible alternative to the use of a Laplacian. And in terms of features, I've already explained how switch cost was generated. The authors used a univariate correlation to relate switch cost to brain features. And their brain features there were mean aligned or liberal signal energy. Just to explain what this means, I've mentioned how it's possible to filter signals using connectome harmonics. So in this case, the authors filtered the signals, retaining only the fraction linked to the first 10 or the last 10 harmonics, so the lowest or the highest frequencies. And then they just computed the energy of these signals and took the average across regions. And that's what they used. Their results were that for the aligned signal contribution, the low frequencies, there was no significant association, but for the high frequencies, there was a significant association between switch cost and liberality from anatomy. The more liberal from anatomy you are, so the stronger you express high frequency contributions, the larger the switch cost. Mathematically, again, feel free to discard if you're not into this. Connectome harmonics were generated from the structural connectome directly through eigen decomposition. And in terms of filtering, you can specify a filter as a diagonal matrix H, where you fill in diagonal entries, in this case with ones or zeros. If you want to keep a connectome harmonic contribution, you put a 1, otherwise you put a 0. To perform the filtering per se, you take your matrix of regional activity over the course of time of size R, number of regions, by T, time points, and you apply the filtering function. So in other words, what you do is that from your original matrix, first you project the data in the spectral domain by pre-multiplying by the transpose of U, the basis of harmonics. Then you pre-multiply by your filter to remove the frequencies that you don't want. And then you pre-multiply by U, the matrix, to go back to the regional domain. You can do this to get an aligned signal or a liberal signal, depending on how you define your filter. And in terms of the extraction of features, I've mentioned that the authors considered the energy averaged across regions. So this is just the mathematical way to formulate this. It's an outer product between the filtered time courses and taking the diagonal of the resulting matrix, because we look at the energy for a given region only and across all regions. And then we just average across the regions to get our final output. Then there was another study done by Alessandra Griffa and colleagues, including some of MIP members, where the Human Connectome Project dataset was considered for 100 unrelated subjects using a larger atlas, the Glasser Atlas, with added subcortical regions. In this case, the modality was the normalized number of streamlines. A population average was still used as a structural connectivity graph, and the normalized Laplacian was used as a graph shift operator. There were multiple behavioral features, as I will elaborate on afterwards. And a multivariate approach was leveraged, namely partial least squares, to relate this set of behavioral features to a set of brain features, which were either standard functional connectivity or equivalent coupled or decoupled functional connectivity, which I'm going to elaborate on. The results of this analysis were that using standard functional connectivity or coupled functional connectivity, the functional connectivity obtained from only the low frequency filtered signals, results were very similar. You can see that the linear combination of behavioral scores, including executive functions, fluid intelligence, or processing speed, amongst others, was pretty identical between both cases, and so were more or less the saliences at the level of the functional connectivity. The R-squared values were above 0.2, so more than 20% of variance could be explained. For decoupling, you see that the pattern, the linear combination of behavioral scores that correlated or covaried actually most with functional connectivity was very different. So there is some additional information that can only be captured in this particular setting with a very, very strong coefficient of determination. Mathematically, again, connectome harmonics were obtained through an eigen decomposition. The filter was specified similarly as before, except that this time the first and last 50 harmonics were retained. Signal processing was similar as well. The only difference is that the author called aligned signal coupled signal and liberal signal decoupled signal. And for feature extraction, you essentially get your filtered time courses and you compute the correlation between the time courses. And that's the quantities that you use. So the size of the feature vector was number of connections at hand, essentially. Then Dong and colleagues followed up on this with an analysis of the same data set with many more subjects, a total of 877, and the same atlas. The exact parameters and specifications for the structural analysis slightly differed, but the important bit is that they also performed a multivariate brain behavior analysis through canonical correlation analysis. And they considered the so-called structural decoupling index, which is another GSP-derived feature. The idea of the structural decoupling index is that you compare the fraction of signal coupled with structure and the fraction of signal decoupled with structure, and you compute the ratio between these two quantities. This gives you the structural decoupling index. And you can see at the bottom left of the slide, the results from the authors in terms of SDI when examining sets of regions as a function of the networks to which they belonged. The SDI values here were computed as log in basis 2. So a value of 0 basically means the same contribution of coupled and decoupled signal. Positive values mean that you are more decoupled than coupled, and negative values that you are more coupled than decoupled. And so the authors confirmed, similarly to the original results from Pretty and Griffa and colleagues, that association regions in the brain and high order networks were more decoupled, while low order networks, such as the somatomotor and the visual ones, were more coupled. Upon canonical correlation analysis, with a very large set of features that included not only cognition-based ones, but also many other characterizations, the author found a significant mode where, in particular, 5 scores reflective of cognition stood out, as you can see on the right-hand side of the illustration. And then they followed up on trying to individually predict each of these scores with SDI and an elastic net regression. They still managed to find significant outcomes, although the associated R-squared values were much lower than with CCA. Mathematically, for the SDI, the idea is that we generate 2 different filters, one to retain the coupled signal and one the decoupled signal. We do processing of the signal in parallel for these 2 contributions. And after that, we compute the ratio between the decoupled and the coupled fractions, as I've explained before. And we get a feature vector of size R, number of regions that we can analyze. Finally, there was a recent study by Siller and colleagues last year, also on the Human Connectome project, with a quite similar set of a population, quite similar choices in terms of structural specifications, but a different atlas, where the authors considered a factor reflective of cognition. The idea is that all the available behavioral scores from the Human Connectome project were preliminarily reduced into only a few summarizing dimensions. In this particular case, 4 different axes, one of which was cognition. You can see this process illustrated at the bottom left, where the cognition score used by the author was obtained from the 4-factor subdivision of the analysis. The authors performed kernel ridge regression and elastic net regression to try and predict this cognition factor from standard coupled or decoupled functional connectivity, as well as from the SDI, to try and understand which of these features would work best on the same benchmark data. And you can see the results at the bottom right of the slide. For functional connectivity and coupled functional connectivity, accuracies were quite similar regardless of the approach that was undertaken. You can see that for decoupled functional connectivity, prediction potential was slightly lower, and that in the hands of the authors, with the SDI, it was even worse. So let us now summarize key observations that transpire from these 4 different studies. And let us try to draw some intermediate conclusions to use afterwards. When you look at the parameters and the choices made by the authors, the only clear commonality really is the fact that all studies considered an average structural connectivity matrix for their analysis. But when you think about it, I would argue that it may be problematic because the functional data is essentially defined on the structure of the subject that you consider. But then you are trying to express it with a structural connectivity matrix that is not the structure of the subject at hand, but is rather an average between all different subjects from your population. So it could be that due to this mismatch, some of the signal that you capture in different frequency bands is not genuine and simply comes from said mismatch. Then, when you think about it, one can understand the use of an average structural connectivity matrix because perhaps you're interested in only shared structural attributes across your subjects, and perhaps you want to discard individual specificities. But then it's also a question whether averaging over a limited set of subjects does the job or not. Could be that it's not enough. And also, when you think back about what I described in terms of a continuum, in terms of brain behavior prediction, it makes sense that you would want to keep inter-individual variability to achieve this, because this is precisely what we aim to characterize. And actually, there are quite a bunch of studies that have used subject-specific connectome harmonics instead, and shown promise in terms of understanding, for example, 22q11 deletion syndrome. Then all of the steps actually differed quite markedly across studies. For example, the atlases used were different, their scales were different, the properties that were considered were different, the graph shift operators were different. In terms of brain features, some studies considered functional connectivity, dimensionality, number of connections, while others considered energy, dimensionality, number of regions, coupling or decoupling with different parameters or structural decoupling index. And then in terms of behavioral features too, in some studies, we consider one behavioral score. In others, we mix many of them together in a multivariate approach. Yet in others, we first derive factors and then analyze the factors. Finally, performance evaluation as well, multivariate versus univariate. In some studies, done in sample. In other studies, out of sample, different sample sizes. So all in all, what can you really say about how different and how well different metrics work? It remains unclear, and you would like to try and remove these sources of variance to draw finer, more accurate conclusions. And finally, importantly, it seems like GSP features do not really do better than functional connectivity for now. As I've shown in the last study that I described, functional connectivity stays on top. And that could be seen as surprising, because when you think of it, you could argue that with graph signal processing, you sort of generalize functional connectivity, because functional connectivity in the standard sense is essentially the whole signal where you keep all spectral frequencies together. So there is an open question. Is it because there is no information of use in the data to leverage GSP tools? Or is it because maybe we don't use the right tools yet and we could make more clever choices? So based on this, there are quite a few research questions of interest that I will briefly touch upon in this talk, but I'm very happy to discuss them more in details for interested attendees later on. The first one is the investigation of potential pitfalls of average connectome harmonics. The second one is to explore whether subject-specific harmonics per se, without even functional signals, simply using structural information, can be useful for prediction. The third is to compare candidate GSP metrics on the same benchmark data with the same choices to limit the sources of variance that affect the results. And the fourth one is to potentially propose novel approaches to improve graph signal processing-based analysis in the context of neuroimaging. So in terms of the data set that I considered for this analysis, it was the Human Connectome Project data. I must acknowledge the precious help and support of Jagruti Patel and Mikhail Schurtner, who were two PhD students who now successfully defended their thesis, who helped a lot with pre-processing of the data, discussions and refinement of some of these results. So I'm very indebted to them for their assistance. In terms of subjects, almost all the analyses used 874 of them, except when I needed to compare scales of the atlas that I considered, in which case I discarded some because otherwise the connectomes were not fully connected. I considered the Lausanne 2018 atlas. The advantage is that you have this same atlas at 4 different scales, so it's possible to investigate what is the best scale for the job at hand. I considered normalized fiber density, compared in some settings average and subject-specific connectome harmonics. And in terms of behavioral feature, I always consider prediction of the cognition factor, like Siller and colleagues, using kernel ridge regression, which has been suggested as potentially the gold standard for such a setting in past publications. And in terms of features, depending on the results that I will show, I touched upon standard, coupled, and decoupled functional connectivity, but also signal energy, as Medaglia and colleagues originally considered. And as I will explain later, also a coupled structural connectivity. First, stability of average connectome harmonics. What I did for this analysis was to compute average connectome harmonics on a certain number of subjects, say 5 or 10 or 20 or 50 and so on. And I did that 100 times, subsampling different pools of subject each time. And then I computed similarity between harmonics after having matched them across all folds and across all cases. You see in the plot in panel A, all harmonics from left to right, from low frequency to high frequency. And each colored curve denotes similarity across folds for a given harmonic, using a certain number of subjects for averaging. What you see is that when more subjects are used for averaging, the curves tend to take higher values everywhere, which makes sense. We average over more subjects, so we get further rid of individual contributions, and we focus on shared attributes. But the important thing here is to notice that even when you use up to 500 subjects or so, you still have quite a few harmonics for which similarity is not that satisfying, which means that if you do an analysis on 500 subjects and someone else does an analysis on 500 other subjects, maybe your harmonics are going to be different simply because of the exact subjects that you chose, which is potentially problematic. So from these results, it seems to me like at least 800 subjects would be a very good choice to make sure that we completely remove inter-individual effects, which is, of course, not easy to achieve. And I think it's worth bringing this forward. And I also observed that intermediate and high frequency harmonics are much more affected than low frequency harmonics. You can see that the first 50 ones show similarity values that are consistently very near 1. Now, what happens when we try to predict behavior with average connectome harmonics? It turns out that you can compute the bias in signal that you get from expressing the data using average connectome harmonics compared to the ground truth harmonics from the subject at hand. Mathematically, I'm happy to give you further details afterwards if it interests you. And the idea is that you can compute functional connectivity, the bias in functional connectivity resulting from this, and coupled and decoupled functional connectivity using either average harmonics or subject-specific harmonics. Here you see on the left side standard functional connectivity in this case. I won't go too much into details, but I would say that it's quite it makes quite a lot of sense. You see that cortical regions on both hemispheres are strongly functionally connected. You have a few negative valued edges as well here and there, and there are overall lower values of connectivity involving subcortical regions like thalamic, hippocampal, and so on. On the right-hand side, you see functional connectivity bias. The upper triangle shows this bias for an indicative subject, where you see that the bias is really present at the whole brain level. And the bottom left triangle shows the average across all subjects from the population, where you see that there is, it seems, a consistent bias across all subjects, specifically when it comes to subcortical-cortical or subcortical-subcortical connections. Now, you can compare all these variants of functional connectivity to predict this cognition factor. First, if we try to compare average and subject-specific basis for coupled and decoupled functional connectivity, in panel A, you see that there are positive correlations in both cases. So to an extent, the results are quite similar. They look quite similar. If you compute this not on an indicative subject, as in panel A, but at the level of the full population on panel B, you see that for the coupled case, the average and subject-specific functional connectivity variants are extremely similar, 0.95 or so. But for the decoupled case, they are more dissimilar. So it seems like there are more notable differences, and so perhaps a bigger impact at the level of prediction. So looking at the prediction results in panel C, you can see that using standard functional connectivity, we achieve an R-squared of roughly 0.15, which is consistent with previous studies. And you can see that using coupled functional connectivity, subject-specific or average basis of harmonics, you also predict significantly, but to a lower extent. The interesting part is that with decoupled functional connectivity, you predict significantly better using the average basis than using the subject-specific basis. And at the same time, the bias alone enables a significant prediction. And so it may be that the reason why prediction is better for the average connectome harmonics case is because it also includes this bias. It also includes this mismatch, and this is partly what drives the results. And this mismatch comes from structural properties of the subjects, not from the functional signal expressed on the structure. I should mention that these results are obtained on the first resting state scan of the HCP, but I also reproduced them on the second scan, although I don't show it here, and they are also significant on the second scan. So it really seems to be something robust. Now, in terms of subject-wise connectome harmonics, if we try to go a little bit deeper and look at what they look like, you have two subjects here for which I show a few examples of harmonics. And you can appreciate, after they have been matched, by the way, you can appreciate that the lowest frequency harmonics, 1, 2, 3, 4, and so on, are very similar across the subjects. The same applies to the highest frequency harmonics. You can also see that they are quite similar, although they are also very localized. A lot of the brain shows values near 0. But you see that for intermediate harmonics, let's say a number 132, for example, the regions that are spanned are completely different. So it seems like there is a lot of dissimilarity. You go from global harmonics that span the whole brain to localized information, as I've said. And as I've just explained, intermediate frequency harmonics are the most variable. If you compute similarity across harmonics, you get a U shape like this. This has been done previously by Cipes and colleagues. And I reproduced exactly their results on the Human Connectome Project dataset, as you can see at the bottom of the slide. But the interesting part is not just to look at the similarity between the harmonics that are matched. It's also interesting to look at the similarity between harmonics that are not matched. When doing so, Cipes and colleagues obtained the following representation, where there doesn't seem to be a lot of structure. In this figure, the diagonal of this matrix is basically the U shape that I described beforehand, and the off-diagonal elements denote similarity between harmonics that are not matched, so that are quantitatively different. When I tried to compute the same on my side, I obtained the following representation instead, where you can see a much clearer structure, especially for low frequencies. Simply put, it seems like there are subsets of harmonics that tend to be particularly similar to each other, although they are not matched. You can see these squares along the diagonal of the matrix. Of course, I wondered why I obtained these results, while Cipes and colleagues did not. And I think it's because of the sign. Cipes and colleagues adjusted the sign of harmonics. Harmonics are vectors with a direction. The sign can be adjusted, can be shifted, but they adjusted the sign only for matched pairs. They didn't consider the fact that for non-matched pairs, the sign is not necessarily right. So on half of the subjects, the sign is in a certain way, and in the other half, in the other way. So it kind of cancels out. In my case, I added an absolute value to account for this, and this is why I reveal this extra structure that really is in the data. And not only is it in the data, but it's actually very important, I would argue. What I did to show this is to approximate structural connectivity matrices only from the first few harmonics. I take either the first 5 or the first 10 or the first 20, and I compute an approximate structural connectome only from this. This is what I called a coupled structural connectome beforehand. And when I did that for all possible numbers of combined harmonics, every time I quantified prediction performance for the cognition factor. And I observed that in some settings, I could actually predict significantly with up to 5% of explained variance. And more interestingly, I've highlighted the starts and the ends of the blocks of harmonics that I described in the previous slide with the vertical orange lines. And you can see that prediction drops and rises quite in accordance with these thresholds. So it really seems like you can predict better when you include all the harmonics that are similar to each other to a certain extent, and that probably together account for meaningful signal. So to briefly summarize up to now, we saw that average connectome harmonics were only robust when averaging over a lot of subjects, and that there is a significant perturbation of behavioral prediction due to the signal dropout when using them. And we saw that subject-specific harmonics can already per se enable significant prediction when carefully selecting which harmonics you include. Now, to conclude, I just want to show you a few comparative results for different graph signal processing metrics on the same data set. So this time, I project resting state data on my harmonics, and I compute For example, in this case, coupled and decoupled functional connectivity at 4 different scales of the atlas. What you can see here is that from scale 3 up to scale 4, the purple and the light brown cases, respectively, prediction reaches a similar level of quality. It doesn't improve anymore from scale 3 to scale 4. So it seems like scale 3 is the best scale to consider. In each of these plots, going from left to right means that you gradually filter in more and more linear manner. You incorporate more and more harmonics for your filtering, by the way. So what you can see is that, for example, for the coupled functional connectivity case, prediction rises as soon as you start incorporating a little bit more of the low frequency harmonics. For the decoupled case, you see that prediction rises much later because you start incorporating the harmonics from the highest frequency ones. So in other words, what it means is that low frequency harmonics are the most relevant ones in this particular case. Very briefly, when you compare functional connectivity and signal energy, clearly functional connectivity wins. I think it's pretty unequivocal in both cases. And I think it may partly be due to the dimensionalities at play. Energy is a regional vector. Functional connectivity is a vector of size number of connections, but this remains to be further studied. And finally, I tried to figure out how to potentially further improve behavioral prediction with first easy tweaks. And one idea that I had was to consider filter weights that were not just binary. So far, I've explained how we set filter weights to 0 or 1 if we want to discard or retain a harmonic, but you are free to set to select any value in the continuum. So I thought, why don't I try to use the U-shape of cross-subject similarity for the harmonics instead? Or perhaps it's inverse. And you can see in these depictions the results where the original binary filter outcomes are shown in green, using the U-shape in purple and using the inverse U-shape in orange. Interestingly, it seems that the latter case slightly improves prediction, which may imply that favoring heterogeneous harmonics across subjects, the intermediate harmonics, may boost accuracy in predictive settings. So to conclude, with a few take-home messages. In terms of average connectome harmonic pitfalls, I've shown you that average harmonics only become truly robust when using more than around 800 subjects. And I've shown that the fact that average harmonics are not exactly the right scaffold to analyze functional activity, there is signal dropout, and it can tangibly alter predictive performance. Subject-specific harmonics are particularly similar across subjects for the lowest and the highest frequency cases. And if you take the right subsets of such harmonics, you can already find behaviorally relevant information. And it seems like a few hundred regions, around 300 or so, is the optimum for behavioral prediction. It also appears like functional connectivity does better than signal energy in terms of cognitive, of predicting cognitive scores. And finally, functional connectivity in these simple approaches where you considered previously studied metrics does at least as well as GSP-related approaches. But novel approaches that go more in depth have the potential to overcome this current hurdle. I've shown an example with this inverse U-shape filtering function, but I believe there are much more potent approaches that could be envisaged. One example, for instance, would be to perform cross-frequency assessments. The structural decoupling index only considers individual regions, but it would be possible perhaps to contrast regions in terms of aligned with respect to liberal contributions, for example. Another thing is that the structural decoupling index doesn't consider temporality because it computes the norm separately for the coupled and the decoupled fractions. So perhaps accounting for temporality could also be a way forward. So to end this presentation, I wish to thank the people who made this work possible over the years. Most importantly, Patrick Hagman, who was my supervisor during my stay at CHUV, and Dimitri, who has always been a mentor and a great guidance for me throughout. Jaruti and Mikkel, who were the two PhD students who assisted me with pre-processing of the data set. Michael Chan for some mathematical discussions that helped me a lot figure some of these results out. Dr. Aleman Gomez for his help with the Lausanne Atlas in our analysis. And finally, the members of the lab where I now work, Shad, Lorena, Miriam, Rupal, and Professor Ingrid Beck herself, as the positive spirit that they've created has also helped me further continue these analyses in my off time over the last few months. Speaking of the future, I also want to conclude the presentation by mentioning that I will actually be ending my contract in 1 month. And so I am looking for potential opportunities job-wise. I've printed out a few CVs. So if there is anyone interested in discussing, feel free to come and grab one. I'm also always eager to just collaborate and discuss and have fun doing research. So don't hesitate to catch up with me at any stage. Thank you very much.